So how exactly does a single solitary swollen vein cause such catastrophic microscopic devastation? I mean, it's honestly one of the most fascinating and frankly frustrating mysteries in reproductive medicine. We're talking about a basic structural plumbing problem, right? But somehow it translates into a total systemic shutdown of cellular production. It's wild.
To really wrap our heads around this, we have to realize that human sperm production is basically a highly specialized biological factory. And like any high-tech manufacturing facility, it requires absolute, unrelenting climate control. Literally. The testicles have to operate exactly 2.5 degrees Celsius cooler than your core body temperature. If that climate control system fails, even just a tiny bit, the entire factory descends into total chaos.
Welcome back to The Explainer. Today, we're diving deep into the specific, incredibly complex biological cascade that kicks off when that precise cooling system breaks down. We are going to explore exactly why varicocele heat kills sperm, and we're going to track this destruction all the way from the physical anatomy down to the molecular and hormonal levels.
Here's our roadmap for today's journey. We'll start with the anatomy of a varicocele, then look at the testicular heat trap it creates. After that, we'll dive into the rise of oxidative stress, meet the molecular wrecking balls, witness the apoptosis chain reaction, and finally, look at the systemic hormonal fallout. All right, let's jump right into part 1, the anatomy of a varicocele, which is basically a massive physical bottleneck.
So what is it? A varicocele is an abnormal dilation and twisting of the pampiniform venous plexus. That's just a fancy term for a network of veins right inside the spermatic cord. If you know someone with varicose veins in their legs, or maybe you've got them yourself, this is an incredibly similar structural issue. The venous valves basically become dysfunctional, so the blood really struggles to pump back up toward the heart. Instead, it just pools there. And this pooling, it's not some rare anomaly. Varicoceles affect up to 40% of men struggling with primary infertility. It's a huge structural bottleneck.
Which brings us to part 2, the testicular heat trap, or what happens when the cooling system breaks.
Now, there is a stark night and day contrast between a healthy system and one with a varicocele. In a healthy microenvironment, you've got a perfectly climate-controlled system. There's this genius countercurrent heat exchange mechanism that uses cooler returning venous blood to actually lower the temperature of the warm arterial blood coming from the body. It keeps things cool, keeps reactive oxygen species, or ROS, low, and maintains an intact blood-testis barrier. But in a varicocele microenvironment, because that venous blood is just pooling and stagnant, that entire countercurrent heat exchange is completely broken. The result is a localized heat trap, hyperthermia. The scrotal temperature spikes, generates massive amounts of ROS, and physically breaks down those critical protective cellular barriers.
Okay, moving on to part 3, the rise of oxidative stress, where we really get into the cellular chemistry.
Allow me to introduce the true villain of our story here, reactive oxygen species, commonly known as ROS. These are highly reactive chemical molecules. Now, under normal, healthy conditions, ROS are kept at very low levels, and they actually help the sperm out. But when the temperature rises inside a varicocele, ROS generation just goes into absolute overdrive. They multiply so rapidly that they completely overwhelm the tissue's natural antioxidant defenses, things like superoxide dismutase and catalase. It is a total biochemical overrun.
When that localized heat strikes, it directly triggers this massive ROS accumulation. And this elevated temperature also forces a breakdown in heat shock factors. Now, these factors are supposed to regulate heat shock protein 70, or HSP70, which is this absolutely critical protective protein that helps cells survive stress. But under the relentless, persistent heat and oxidative stress of a varicocele, HSP70 just breaks down. And without its protection, the cell initiates caspase signaling. That's basically the biological equivalent of a self-destruct sequence, leading directly to cell death.
Let's look closer at that damage in part 4, the molecular wrecking balls, because this damage is completely indiscriminate.
These reactive oxygen species are literally molecular wrecking balls, and the collateral damage is devastating across 3 specific cellular structures. First up, the DNA, the cell's actual blueprints. ROS physically attack the guanine bases in the DNA, causing severe mutations and fragmentation. Second, the lipids, which act as the fortress walls. ROS actively destroy the polyunsaturated fatty acids that make up the sperm's outer membrane. And third, the proteins, the factory machinery itself. ROS trigger a process called carbonation, which makes critical structural proteins misfold and ultimately fail. To put that lipid damage into perspective, sperm membranes are incredibly rich in polyunsaturated fatty acids. They need them to stay fluid and flexible. But oxidative damage triggers something called lipid peroxidation. This process literally rusts the fatty acids. It tears actual holes directly in the cell membrane and releases a highly toxic byproduct called malondialdehyde, or MDA. Once those fortress walls are breached, the sperm completely loses its structural integrity and its ability to function.
And the damage doesn't even stop at the surface. Inside the cell, ROS are attacking the machinery itself through protein carbonylation. Think of carbonylation like throwing a giant physical wrench right into the gears of a microscopic engine. The ROS chemically alter the proteins, which leads to widespread errors and changes in the protein structure. Most critically, it shuts down mitochondrial function. The mitochondria are the batteries of the sperm. Without those batteries, the sperm has absolutely zero energy to swim, let alone actually fertilize an egg.
Which brings us to part 5, the apoptosis chain reaction, or programmed cell death. When you piece all this forensic biology together, you get a very clear, step-by-step path to cellular ruin. It always starts with step 1, localized heat accumulation from that stagnant pooling blood. That forces step 2, the massive, uncontrolled generation of ROS. Because the cell's blueprints, walls, and machinery are taking such catastrophic damage, the cell realizes it's beyond repair and triggers step 3, caspase activation. Caspases are essentially the executioner enzymes of the cell. Once they're activated, they initiate step 4, programmed cell death, officially known as apoptosis. The damaged germ cells are literally orchestrating their own demise.
But the tragedy here isn't just the loss of individual cells, it's the total collapse of the local infrastructure. The extreme heat and ROS attack the tight junctions between Sertoli cells, completely breaking apart the blood-testis barrier. When this barrier is disrupted, developing germ cells physically detach from their life support. And here is where it gets truly shocking. That barrier? It's supposed to hide sperm from your body's immune system. Once it breaks, your body's own white blood cells flood in, treat the sperm as foreign invaders, and launch a devastating autoimmune attack. No way around it, it's brutal.
Finally, we reach part 6, the hormonal fallout. Because this disruption is system-wide. The hostile environment created by the varicocele doesn't just kill sperm, it actually poisons the surrounding support cells. Leydig cells, which live just outside the tubules, suffer impaired stereogenesis. Basically, they lose their ability to synthesize hormones, causing a drop in internal testosterone levels. Meanwhile, the Sertoli cells, the nurse cells, are damaged and can't offer as much support. Because this vital interaction is broken, they stop producing a hormone called inhibin B. Without inhibin B acting as a regulatory brake, the pituitary gland in your brain absolutely panics and pumps out elevated levels of follicle-stimulating hormone, or FSH. The whole hormonal feedback loop is just completely thrown off balance.
And this entire system is remarkably sensitive to temperature. Like, why exactly does testosterone drop? Well, heat literally turns off a single, highly specific enzyme called C1720 liase. Think of it as a super delicate microscopic switch. Without this switch flipped on, the body simply cannot convert precursor hormones into testosterone, so production just stops. A tiny, localized spike in temperature from pooling blood turns off that biochemical switch entirely. It is a stunning example of how a physical heat trap dictates chemical realities. So we've seen how just a few degrees of heat trapped by a faulty vein can unravel DNA, tear apart cell membranes, and shut down hormone production. But it leaves us with a critical, much larger question to ponder. If a localized, microscopic heat increase causes this much biological destruction, how else is modern life quietly turning up the temperature on human fertility? Think about our daily habits, our environments, the laptops we rest on our legs. What other invisible heat traps are we walking into every single day? Definitely something to think about. Thanks for joining me on this explainer, and keep questioning the science around you.